Okay, so this is going to be a talk about how you can do distributed computing with biology. So I realize that this might be a kind of outlier topic. So if you have any questions at any point, please feel free to interrupt. Um, More or less, I'm going to introduce the concept of what it means to compute with living matter, with bacteria, with cells. And then kind of go into like a modeling section where we try to understand how we can understand these things mathematically, how we can reason about these things kind of in quote unquote our language. Then going to kind of a more like practical things, like I'm going to talk about what you can do locally, what we understand we can do locally inside of a bacterium, how we can change it, how we can program it. And then in the end, I'm going to go into like how we can use these local algorithms to do something on a population level, like real like distributed algorithms that we are going to program. So first of all, what are we talking about? So we're talking about living matter. So what is life? Life is everything that goes on inside of cells. So cells are compartments with chemistry going on inside of it. Right, so lots of proteins, but also other molecules that are reacting together. And so by definition, life is inside of cells, different compartments. That means also by definition, it's a distributed system. Like the quality of this is that all the molecules from one compartment cannot go into the second one and interact with it. Maybe with intermediate steps, but not directly. And so here you have a picture of a bacterium. Bacteria are like the simplest forms of life. That's why people study it. The most studied is the E. coli, the Escherichia coli that you see here. It's a rod-shaped bacterium. Mostly when people do experiments, that's what they use because it's the most used, most understood, one of the simplest bacteria also. Sometimes, of course, you see them in the news, like when there's like an E. coli infection, like in a supermarket or something. Not all of the E. coli are pathogenic. Not all of them are bad. You have lots of them in your gut, and they help you, of course. So they kind of just to give you like a more or less an idea of how big these things are. They kind of 2 micrometers long, half a micrometer wide, rod-shaped. And these, of course, have like inside of them, they have lots of stuff. In particular, they have a genome, like we do. They have DNA inside of them. Kind of the DNA kind of encodes the proteins that are expressed. More than that, also, but in particular, the proteins, and you can see that kind of as the source code of the bacterium. And this is something that we can change. And I'm going to talk about like how you can change the source code of a bacterium. They can interact with the outside world, so they're compartments, but they're not completely sealed off from the outside. So they have like these E. coli in particular. They have this double membrane thing going on. So on the top here, you have the external world. On the bottom, you have the internal world. In between, there's also some things going on. There's like the periplasm between the two membranes. But this is just to say that kind of these membranes kind of seal it off a bit from the outside, but they're not 100%. There's some things that can go through that. In particular, they have these receptors that kind of attach to things from the outside world. So when of course now, like everybody heard during COVID, like that there was this receptor that's on the outside of some cells, and kind of the virus tries to find them and then interact with it. It's not just for viruses, but in particular, that's what happens. Right. So the you can see like these cells as yeah. Oh yes, I forgot to say. So bacteria are single cellular organisms, right? So we of course we have many cells, but like they are just one. Yeah. Right. So you can see kind of a cell, a bacterial cell, kind of that's the whole organism that there is as a system that kind of takes in some inputs and produces some outputs. Like inputs might be light metabolites, oxygen, like the pH level around it can be seen as an input. Like sugar, they like amino acids, whatever they can get. And so in a very abstract way, you can see kind of you can have inputs and outputs, and then between there's things that define how we go from inputs to outputs. Of course, the genome is the most important one. And the thing is that, as I told you, we can't change that. So of course they have the genome, and they replicate the genome whenever they replicate, whenever like one mother cells duplicate into two mother cells. Like that's how bacteria duplicate. They replicate, of course, the genome, but we can't put more into the cell than they have at the beginning. So what you can do, and this is kind of this field of synthetic biology, has been going on for more or less 25 years. What the idea is that you construct a piece of DNA. How to construct that, I will tell you in a second. But once you have the piece of DNA, you put it inside of the cell. You put it inside of the cell just by forcing it into. Like you can heat shock it, you can electroshock it, you can chemically shock it. What that does with the bacterium is that kind of it opens up a bit the membrane, and you hope that some of the DNA floats inside of it, and then you hope that it lives. And some of them will. Many of them will die. Many of them will not take up the DNA, but some of them will. And you can figure out who got the DNA and who did not get the DNA. And once you have that, then now you have a bacterium with its own DNA and some of the DNA that you put inside of it, right? And kind of the mechanisms of the cell, of the bacterial cell, it doesn't know what is what. It doesn't know that this is the DNA that kind of, it's its own and that's the other DNA. It will just express whatever you put on it. And in particular, you can put on it, like I was talking about replication. You can put on it like a substring of the DNA that tells the cell to also duplicate this piece of DNA whenever it duplicates. It's known kind of, people have figured out what that is. It's a sequence like DNA is a sequence of 4 different characters, GACT. Some of the strings are known, and this is one of them. And if you put that on, then kind of the daughter cells, the lineage will have this plasmid, so this piece of DNA, which is called a plasmid. Okay, and then you did it. Now you have introduced into the cell a new piece of source code. How to construct this DNA in a very simple sense? You can, or in a simple way of looking at it, you can just get like a string, GACT. You can write it down on your keyboard, copy paste it to on a website of companies that sell you DNA. And next day you will have it delivered to you. You should put it into a freezer, but that's more or less all you need to do. And then now you have the DNA. Yeah. So there are size constraints. So you cannot, like, for example, you cannot put human DNA. You cannot. It's much too big. You cannot just order human DNA. Even the whole genome of a bacterium, like a second genome, you couldn't order. It's too big. They don't sell you that. What you can do, though, is take pieces of DNA that you already have and edit it. So there are DNA editing tools available to you, restriction enzymes, CRISPR, which like got the Nobel Prize a few years back, and then you can have then you order just a little bit that you want to change, and the rest will remain the same. Yeah. But like yeah. I will. Yeah. I'll say it's an excellent question. Yeah. Okay. So in principle, now you can introduce DNA inside of your cell. So you can kind of have it express more genes than it did at the beginning, and other things. It can do other things as well. So let's see some examples. So here you see a microscopy image, which will soon turn into a microscopy movie. Each one of these rods is a bacterium. Of course, this is like a time lapse, and you see kind of that they grow, kind of they duplicate. There are few of them, then they grow, kind of duplicate, and then you see these flashes. And then once these flashes happen, like you see many of them just being gone. There are a few things going on here. So this is kind of a distributed algorithm that is implemented, that like these bacteria do communicate with each other. And what happens is that kind of there is a specific mechanism that is understood and that bacteria have figured out, which is called quorum sensing. They can kind of sense how many other bacteria are around them. And if enough of the bacteria are around them, then they can do something. It's used for multiple different things, but for example, it's used in the natural world for biofilm production. So kind of bacterial colonies, they can protect themselves by producing biofilm. But if there's just one bacterium, it's not worth the effort. If it produces some of the biofilm and nobody's around, it would just get lost. But if many of them are around, then it makes sense to express that. And so they figured out this quorum sensing mechanism. And this is what has been implemented here. Like this is a synthetic circuit. This is piece of DNA. It has been like copy pasted together with known genes, copy pasted into web form and ordered. And what it does is, if bacteria see that there are enough other bacteria around them, they will kill themselves. They will light up green and kill themselves. That's all happens here. That's all that happens here. What the effect of that is that you have like this oscillation pulse going through the system. And it's kind of, at some point it gets lost, but during a certain amount of time, it's a nicely a synchronized clock. And the synchronized clock, like during not the whole population, but a large part of the population. Yes. So you put a gene inside of them that will kill themselves. Yeah. Yeah. So the flashes are very specifically, this is what's called a gene fluorescent protein, GFP. Which is kind of the state-of-the-art output reporter of a bacterium. So what it does, like two things happen whenever the quorum sensing mechanism is triggered. So it sees that there are many bacteria around it. It will first flash green. So it will do two things: flash green and express the lysis gene, so the killing yourself gene. So both of them. That when it flashes up, you can expect that the bacterium will die. So it's just to be able to measure like more easily like what the flashes are. You make them light up green, and you have like this measurement device that can kind of very well read these green flashes. In the new DNA, yeah, you wouldn't have that. Yeah. It can be. So this does come from a virus that detects bacteria, and like people have figured out what the gene is, and they copy pasted it onto the new DNA, and they introduced that. Definitely, yeah. But what you're referring to is like phage phage therapy, which is also absolutely a thing. And it's the same kind of gene that's used here. Yeah. So why would you do that? So first of all, it's nice to look at, but it also has applications. And then in the same paper, actually, the authors here gave an application, a medical application. So these poor mice, they get mistreated sometimes, and it is the case here. So there were mice where they introduced a tumor cancer, and then they, of course, like on their own, they would not have a very long life expectancy. But then they figured out like this. They used their mechanism, this flashing mechanism, flashing and killing themselves mechanism, the synchronized clocks. They used it to deliver a toxin to the cancer. How does that happen? So first of all, like now they put another gene into the bacterium that I did not tell you about yet. So they had the green flashes, the killing yourself, and then also the toxin. All three of them are produced. The toxin gets released when the bacterium kills itself. It opens up the membrane. Like the killing itself is by destroying the membrane. Once the membrane is destroyed, the toxin gets out. And the hope is to kind of attack the cancer with this toxin. And it turns out that if you do it in flashes, it's more effective than if you do it like an average over time. It takes the same amount, but it's just concentrated in flashes. It turns out to be more effective. And so why doesn't the toxin kill the whole mouse? It turns out that this kind of like by chance or by happenstance, it turns out that in cancers, in a tumor, the immune system is much weaker than outside. So bacteria, like foreign bacteria, have a higher propensity to be at the tumor site. Kind of, so that the authors didn't have to do anything for that. Kind of by nature, that's where they go. And then they showed that kind of a higher life expectancy of their mice. Most of them still died. That's how medical research goes, unfortunately. But yeah. Yeah, and they also showed that kind of in combination with chemotherapy, it's even better than if you just do this. Yeah, and this is kind of a field of application that kind of is very promising in my view. We'll talk about maybe at the end, but kind of you oh, sorry, that's a question. Yeah. No, no, yeah, it's a very good question. So does the immune system killing them also release the toxin? Well, kind of a small amounts, but you need to have these colonies building. So if there's just one bacterium, like it just gets killed, maybe there's some toxin released, but that's it. But you really need these large colonies. Yeah. Yeah. Yes. Correct. That's the whole idea. So if you had like another device that ran on a clock and released this toxin, that would also be fine. The advantage of bacteria is that they can live like in our bodies or in mammals' bodies or other animals' bodies. Oh yes, yes, yes. So we saw that in a movie just here. So most of them get killed, but some of them live, and they rebuild kind of the colony. So yes, so if everybody was killed, like you would get one flash and that's it. It turns out that you have multiple, and more or less like you can see that, and we see that it's a theme in biology that everything is noisy and stochastic. So kind of you want to kill everybody, but not everybody gets killed. Yeah. Yes. Yes. Oh, you absolutely can do that. So kind of you could also like stand next to the mouse and just inject it like yeah, you could do that. You could do that. What's that? It depends where the cancer is. If the cancer is like at the site that is accessible, you can do that. Yeah. It turned out here that it would have been possible here. Yeah. Oh yeah, yeah. So you can think. So this is of course like people are not really interested in curing cancer in mice. Of course, they are interested in curing cancer in humans. And there are lots of like tumors, cancers that are not accessible. Like one that's yeah, many of them aren't. Like pancreas and other areas, it's really really badly accessible. So. Yeah. So it's not about cells, it's like parts of the body. So parts of the body where kind of you have like an active immune system. It's not just one cell that has an immune system. Many of the large parts of the immune system are cells. But many of the parts where there's no tumor, like you have an active immune system. So if you have some of the bacteria that are like pathogenic, they will get rid of. But in the tumor, and it's not just a question of one cell, but like a collection of cells, there the immune system is much weaker. So we'll have these colonies forming there. Yeah. Yeah. Yeah, yeah. So yeah, just to wrap this discussion up about like possible applications. So this is one maybe one of the more promising things where computing with bacteria sounds really exciting because you can, for example, like cancer therapy is one, cancer detection is another. So you could think of like having some of the many trillions of bacteria that you have in your gut, just doing readouts about your health. Like for example, like trying to detect free floating cancer DNA, which happens if you have a cancer, some of the DNA just floats around. And maybe like you could like detect some cancers earlier than you would be able to otherwise in these kinds of things. So there's some exciting applications you can think of. Yeah. Like timeline. Your guess is as good as mine. Yeah, yeah. So yeah, you can think of other applications of like changing the source code of a bacterium. Some people try to do that or think about that as using that really as a computational resource, like more or less like doesn't need electricity. It just needs like a cube of sugar and then it does something for you. I think that's less exciting for them. Okay, so let's get an idea of about scale. So here we have two computing machines. We have like IBM processors and an E. coli. So it's like a 3 nanometer process, pretty much state of the art. And then just if you compare kind of the volume or the area here rather of an E. coli, you would be able to fit more or less like 200 transistors into one E. coli on the area that takes up one E. coli. With that, you could build for more or less like 50 NAND gates. So of course, the bacterium does much more than that. So that's kind of just like if we took our like state-of-the-art electrical circuits, we could do 50 of them. In the bacterium, there's much more going on. So here's this just some of the pathways of what the bacterium actually computes or produces or whatever, like things that it does. Of course, not all of that is usable. Many of these things are necessary for like the upkeep of the cell. But just to say that kind of in terms of scale, there is like large potential there that's kind of untapped, of course. It is just another picture of scale. This is more or less like a map of the genome of E. coli. So what's going on inside of these bacteria? So I told you that's kind of there's chemistry going on inside of these compartments. The compartments, like there's chemistry going on. And here's like a very nice simulation from now um, with kind of some of the molecules and some of the reactions that happen inside of a part of an E. coli during 15 microseconds. So kind of some people have this image, and I sometimes have that as well, that kind of you have these compartments and they're like, and we come back to that there's like these molecules flying around at some point they meet and then they react or something. Actually, it's a very crowded space in there. So if we want to compute with them, like there are some challenges that we face, of course. It's not as easy to program with bacteria as it is like on a computer like this here. Yeah. Yeah, that's a good point. So what do we want to do? So like think the end goal is like to be able to construct these living machines that kind of can help us. Now, how do we get there? We have to predict or reason about more or less what they can do and what we should do to them so that they do for us what we want them to do. For these things, there are like many things that are thinkable. You can simulate them, you can write proofs, you can do experiments, and everything is fair game a priori, right? So if some of these like lead you to a system that's useful, all is good. Of course, here I will present like a mathematical angle, but of course there are experimental angles that I won't be going into. Yeah, I think in the best 100%, 100%. Yeah, so it's a not very well formalized space, 100%. And I think it needs to be like a dialogue with biology, between biology and the mathematical reasoning about it. Just like one-way streets don't work at all, like based on my experience, but of course others. Yeah, of course. Yeah, 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 yeah. Yeah, yeah. Yeah, yeah. So they do transform, but here it's like more or less the structure that transforms. So like one thing that's very particular about like life is these macromolecules. It's huge, huge molecules, like chain molecules, like DNA, very long chains, RNA, very long chains, proteins, very long chains. And like for example, if you think about proteins, which many of these things are, is like you have these formations and they can change a bit. So like some parts of the your molecule do change, and they produce mechanical force on the other parts, but like it's not the whole molecule that gets like split up or merges with another one. Also, but the macromolecule is not necessarily it's a new entity, but like it can also be a new formation of the same entity. Okay, so challenges. So if we like come to this kind of new world of programming bacteria, kind of, and we do know about electronic circuits, what are the challenges? So here's of course a comparison. On the top, there's a SPICE simulation of of an inverter, and on the bottom, there's also a simulation of a biological inverter. I will tell you about like how to implement these things in a few minutes. But of course, you see that kind of, and sorry, the blue ramps here is the input, and the orange or red is the output. So you see, of course, first that like here there's a ramp in the electronic circuit. Even the output of the electronic circuit is not completely like straight line, but of course, like it is kind of more or less nicely behaved. If you go to biology, like many other things happen here. So stochasticity is very pronounced. It's not the only thing that's going on here. So here, if you look at the time scale, you see 250 minutes. More or less, E. coli, they duplicate every, let's call it 20 to 40 minutes in good under good circumstances. In particular, what we see here is that we have many generations. So we have around 10 generations that we're seeing here, actually. So it's not just that inside of one cell there's noise, like one cell is like takes this trajectory, the other cell takes the other, but in between there were different cells being born. And this, of course, yeah. Yeah. So here it's protein count. Um, the GFP, the green fluorescent protein counts, the things that you can measure. And this would be like a global measurement because like if two, like the superimpose, if one bacterium is lit up with intensity 1 and the others with 2, then you get the readout 3. Yeah, yeah. Yes. Yeah, very good question. Why the many orange lines? This is stochasticity. So there are multiple simulation runs here of the same system, but it's not deterministic. It's stochastic. One time you get that, the other time you get that. Yes, exactly. Different lines are different simulations. And they have some trend in common, but it's not like they can diverge quite drastically. Right, and then there's maybe as a corollary to that, or as another observation, there's like a huge cell variation. So here, of course, we looked at kind of the readout of a simulated bacterial colony. So many bacteria at the same time, but they're not all homogeneous. If you zoom into them, like some of them, so if you look all the way on the right, there's a fluorescence. It's YFP, it's yellow fluorescent protein, but it's the same idea. It's a readout. But you can also look at kind of what the distribution is among cells. And you see that there's a mean where like most, or like where there's a large likelihood of cells having this certain fluorescence, like readout, but there are also some that are lit up like very little and some that are lit up more. And it's just a feature of what that happens. Like we can even have some subpopulations. Like here, you more or less you have one population that's centered around like a mean. You could also have two different means, or three, or whatever. Okay, so how can we reason about this? First of all, we are thinking about inside of a cell before moving on population level. It will turn out that we will reuse the inner cellular model also for the population level. As I told you, what's going on inside of a cell, on a basic level, it's chemistry. So if you go to the models in chemistry, you will see kind of this formalism that's called chemical reaction networks. That's also sometimes pops up in our community. And more or less the basic question that it's supposed to answer is, like, let's say you have a box and you have two molecules, A molecule and B molecule flying around, and you want to know, like, when do they react? Like how frequently do they interact? And let's assume for simplicity that kind of we only have two of them. Let's say there are two balls. Let's say on the plane. Let's pick both of them of radius r, and let's like have them flying around in a uniformly probabilistic sense. Constant speed, but different velocity vectors, like the angle can be different and the initial position can be different. If you only have two of them, like floating around, you can reduce the system. You can reduce the system to, like, first of all, when they touch, it means that kind of the distance is the sum of the radii. So you can kind of reduce one to radius 0 and the other like having 2r now. And it's kind of, you get the same hitting distributions. Yeah. For mathematical convenience. Yeah. So for mathematical convenience, I'm trying to make an argument like that what the kinetics should be, and then we see kind of how that works out. Yeah. So this is for mathematical convenience. So first of all, reduce to 1 point, and let's say only have the second one flying around. Well, as you can add the speed to the other one, or square root 2 of the speed to the other one. And let's say the other one like stays in the middle. And maybe I will use the order. You can see kind of this is a box. In the middle, we have like this 2r ball. And then we have a second one floating around here. And you might assume, which is true, like if you have a box that it kind of it bounces off the box. But if you have exactly the one ball in the middle, it doesn't really matter whether you bounce off the box or whether kind of you come in from the other side. Kind of it's just you mirror every time you hit the ball, you mirror, but it doesn't matter if your target is exactly in the middle. So kind of you can look at this as like just infinite space, infinite repeating space, and you have multiple of these balls. So in particular, it means kind of the hitting probability at some point in time, it's proportional to the radius of it, because you can see kind of at some point in time means kind of you're building up this radius around you. You see kind of what is the surface, the attack surface of your ball, and you kind of have them repeating, but it only depends on the radius and not the area. Okay, so with these simplifications, we can look at, and this is what we want, of course, the probability of like what is the probability to hit this ball in the middle in the next t time steps. Okay. And it would be easy if the hitting time like would not depend on the past. Then it would be Markovian and everything would be nice. Turns out that it's not exactly Markovian, because more or less what you do know, like if you look at the probability of hitting in the next t time steps, given that you have not hit it in the last s time steps, you do know that kind of you're not close to it, like in a phase space, you know that you're not close and having a velocity right point, velocity angle pointing right at it. So there's some shadow phenomenon around it where you make a mistake. Like you do know that you're not there. In a certain limit, so in particular, you can show that if R goes to 0, like the radius goes to 0, so that you have 2 points more or less trying to hit each other. This goes to 0, and you then get equality here in the limit. So kind of that means that it's historyless. Like the probability of hitting in the next T rounds does not depend on the past. And then you're Markovian, and there's only one Markovian distribution. It's the exponential distribution. Right. So kind of just based on the first line here, you do know that what's the probability of hitting in the next T, or like waiting for longer than T, and then you get the probability of hitting T. So that means that you're a Markov chain. It also means you're a continuous time Markov chain, because here we're talking about continuous time. Okay, so let's go back. So we figured out for A and B kind of how the probability distribution should look like for them to meet. There's e to the power of minus lambda t. Lambda we don't really know, but we're going to figure that out now. So let's say you have now not one A and one B, but two A and one B. Yes, please. Oh, it definitely has. Yes, it does depend on the size of the box. It does depend on the radii, but just based on the fact that there's only one memoryless continuous distribution, which is the exponential one, you do know that it's of that form. You don't know lambda yet. And lambda will have the information of how big the box is and everything. And not only how big the box is, but also like if you're introducing multiple copies of one molecule that's flying around, you also do know that kind of if you're now having two of them, now kind of you could double your rate, because more or less you have now 2 times the radius of a text surface. And in particular, and here where the box comes into play, so now if you're reducing the area by a factor of 2, you're increasing the likelihood of them meeting, also by a factor of 2. You can make the calculation. So all of this, that kind of you do depend on the number of A's, number of B's, and the total volume, or like the inverse of the total volume. If the volume gets smaller, you're going to hit each other more and more often. Yeah, and then this kind of reduces down to like things of that form. So here is one chemical reaction in an abstract way, in a formal way. So you have like here, you have A and B, and when they meet, they give C and D. So it means like kind of A transforms into C and B transforms into D. That's how you can think of it. And kind of the reasoning that we just made, it's like this rate of interaction, so lambda should be linear in the number of A's and the number of B's, and it should be linear in one by the volume, or the area in our case. Yeah, and then you can kind of write down formally. This is how those things are written down. So A plus B gives C plus D with a parameter gamma here, and this parameter gamma is encodes a few things. In particular, it encodes kind of how fast they're going and how likely they are to react once they meet each other. And kind of a chemical reaction network. So this is one chemical reaction. You would write some of the chemical species on the left, and then an arrow, some of the chemical species on the right. Those are the products. And the semantic is like you take away of the system when the reaction does happen. So if A and B meet, you take away A and B and you place in C and D. And a chemical reaction network would be multiple of these things going on at the same time. They come into a few flavors. So here we talked about binary reactions. So A plus B here, like giving C. And here kind of our reasoning went that kind of the reaction rate lambda should be linear in A, linear in B, and linear in 1 over V. So it kind of should be like A times B divided by V and some constant. And this gamma that's on top of the error here is what we call the rate constant. And this tells us like how likely they are to interact in some sense when they meet. Yeah, and the dimensions here should be volume over time, more or less, because like lambda should be 1 by time. It's a rate. So that's what you get. These are binary reactions, so A and B meeting. More or less, there are only two other things that we need to consider: unary reactions and nullary reactions. Ternary reactions, quaternary reactions are not really considered in the literature so much, because more or less like what a ternary reaction would mean is that the three molecules have to come together at the exact same time. They have to meet at the exact same time in space. It's very unlikely. And actually what would happen more likely is that first two of them will meet and then they will meet the other. So they will form a complex first and then the other. So you would have two binary reactions instead of a ternary one. So kind of the only things that we need to consider is binary, unary, nullary. So a unary reaction is something like A going to 2A, for example. It's a duplication reaction. So that does happen. Like here, the gamma does have a different dimension. More or less, how the rule goes is like you're taking one. So you always know that kind of lambda should be a rate, so 1 by time, and it should be linear in all the reactants. Here there's only one reactant A, so kind of it's just gamma times A. And null reaction is just like something being produced out of thin air. Most likely that's kind of an in-stream from the outside. You're modeling something that's like an external inflow, external production. Okay. And yeah, then kind of this is what we think about then with this chemical reaction. So we'll have like a list of chemical reactions, and what you will have in your head is like this kind of collection of molecules that are floating around, and whenever they meet, they do something. And of course, the assumption here is there's no space here. Contrary to what we saw in the video, like we saw that there are some flashes, but they're localized. Here, we assume that everything is well mixed. We assume that maybe we stir or they're mobile or whatever, and they're atomic interactions. So kind of on the time scale, much quicker than the movement of the molecules. And once we have that, now we can talk about executions. And there are, like I will give you two different ways of looking at it. There are two different ways of looking at the execution, one being considered more precise, which is the continuous time Markov semantics that we have been talking about. So let's say we have like one chemical reaction network that with two chemical reactions. So A plus B, when they meet, they can form a complex C, and A can duplicate. Maybe it's a bacterial species. That's what bacteria do, they duplicate. Yeah, this is just like how you would, like if you know continuous time Markov chains, this next minute is not for you, but more or less like what you would also always have, like you would have a state. The state here would be count of A, count of B, count of C. A priori they're unbounded. So this is a state. And then you have like these different reaction rates that can happen, and they can happen at the same time. So we have two different reactions. We kind of figured out what the lambda should be for each of them. So the first one would be alpha times A times B. And the bottom one is a unary reaction, so it's just beta times A. And now they're happening in parallel. And whichever comes first, this is the one we take. And then we restart the system because everything is Markovian. Everything is memoryless. So we can restart the system then. So what happens, like each of them is exponentially distributed, like the exponential distribution. Here it turns out maybe that the bottom one is smaller. So that's the one that happens first. So we pick that one. We throw away the other. Now we're in a new state. Like now A got duplicated, so now it's A plus 1. And now we do it again on a new state. And maybe now the top one is the smaller one. And we go again. And because of the memoryless property, like we don't have to look at other things except our state. So this gives you kind of these different curves that we saw here a few slides earlier. So every time you start the system, you get a new trajectory. It's a stochastic process. There's another way of giving meaning to like an execution, a semantic for the same system, which is based on ordinary differential equations. It's considered to be less precise, deterministic, and it's sometimes it's not exactly true, but you can think of it as like of having the mean value, the expected value of all the trajectories. And there's a very mechanical transformation going on here. It's very simple. Now like I write this A in brackets. That means the concentration. So dimension is 1 per volume. So, because now we're not talking about counts, we're talking about concentrations because now we want to have like a continuous variable. And these concentrations would give us that. Both of them are used. And we will use both of them in this talk. There's an obvious comparison that has to be made with population protocols. Population protocols are also about things that meet in the changing state. If you think of them as like under a stochastic uniform scheduler, which many people do, you would have the population protocols being a subclass of these stochastic CRNs, right? And they are restricted to only having binary reactions that produce binary outputs. So A and B meeting, giving C plus D. But it couldn't do like A gives 2A. That's not allowed. Or A plus B gives nothing. Or A plus B gives only C. And also like all rate constants would be equal. So which is in a chemical system is very unnatural. So maybe by chance you would have like only 2 inputs, only 2 outputs. That can happen. But having all rate constants be equal is very unlikely. So some of the kind of flavors or some of the techniques, of course, from population protocols do carry over. We see that hopefully at the end. But they are two different kind of games that we're playing. Yes. Oh, I don't know exactly, but the more or less the idea is that you can have a population, and you can think of them as individuals walking around, and whenever they meet, they can talk and change their internal memory, so changing the state. Why specifically they came up with this name, I do not know. But it is a model that's heavily used in the yeah. Okay, so that's a model. So let's go through one example here. Let's say we're picking, and now we're going to introduce some notation here. This is a gene. So let's say here, this is the DNA. The blue line is your DNA. And the gray box with Z on top is a gene. So like something that codes for a protein. That's mostly what a gene is. In front of and in the back, the two things. In front, there's something that's called a promoter. We will come to that in a second. But this is where kind of the expression of the gene starts. It's a certain subsequence of DNA, like GACT, whatever, that is recognized by the cell's mechanisms to see that, oh, here I have to start expressing this gene now. And at the end, there's a terminator to know when to stop. So let's say we have this gene, and let's say it's always on. So kind of it's produced constitutively over time. And let's say our protein can also be degraded. So we will have two different reactions here. So kind of Z, kind of our gene or copies of our gene will give Z plus O. We're not going to model exactly how that happens, but let's say just the presence of C makes it such that our output O is being produced. O is our protein. And O can be degraded. So O goes to nothing with some rate also. Let's just try to figure out how like the kind of quote unquote easy coarse-grained model would handle that. So ODEs. So how the rule goes is like you would have one equation for each species. You have two species here, Z and O, and you would always like do the derivative of one of the species. And you see, then you're looking at kind of what the delta in each of the directions is. So here, for example, we see Z not appearing in the second one, but in the first direction it does appear, but it appears once as an input and once as an output. So when that direction happens, Z doesn't change. Like we're taking away one, but we're putting back one. So the derivative here is zero. It's a constant. O is a bit different. It appears in both of them. And once it's in the top direction, it appears as a product. Like doesn't appear on the left-hand side, but it does appear on the right-hand side. So it's going to be produced. So there's a plus sign, implicit plus sign after the equality. which is kind of just the rate of this reaction. The rate, we already know this is a unary reaction, so it's beta times z. And then like, but that's not all that happens. Sometimes it's also being removed from the system. And this is just like the minus times the rate of the second reaction, which is gamma. It's also a unary reaction, so gamma times. Okay, so as soon as you have that, kind of you can like integrate this ODE, the release. It's a linear ODE, and if you like assume that z, that O starts at 0, you see this kind of charging curve. There is a limit because at some point, kind of there's a degradation that kicks in if you have too many of them, but you have this exponential approach to kind of your steady state. And I might skip this one. Okay. I don't know exactly when we should do the break. In 10 minutes? Okay, then I go over a few slides. Oh, thank you. Okay. So now let's talk about like how we can use these models and kind of use parts of what's already happening in nature to implement our algorithms. First of them local, then the second step, I will go like population level. So what you can do with genes, what you can do with genes is nature has figured out ways of turning on a gene and turning off a gene in a very simple way that's called like a activation and repression. So here there's both of them going on. A, of course, is activation and R is repression. And more or less, let's focus maybe on the right-hand side. So I told you that in front of the gene, like the gene X here, the yellow box, that's this promoter. And this promoter is needed for the expression to start. And this, I think I have a slide about this, and yeah, here, maybe let's look at that. So here, it's a bit different kind of thing, but it's still the gene. Now it's a gray box, but here's the promoter. And under normal conditions, what you would have is kind of the mechanism of the cell. It's called RNAP. I will maybe talk about it in a second. But what it will do is kind of, it will recognize this sequence and chemically just bind to it. And then once it's bound, it will know kind of that now it can go forward and it can produce RNA out of your DNA. By the way, of course, this is the cycle, what's called the central dogma of biology. You have proteins being produced, and it always goes DNA, goes to RNA, goes to protein. So first, if you want to produce a protein, first you have to do what's called transcription, like changing DNA into RNA. And this is like a one-to-one process. You go from GACT to GACU, but it's like one character per one character. And this is what happens here. So what I want to say here is that like this RNA, like RNA production here is necessary for afterwards the gene being produced, the protein being produced. And now what you can do, and nature has figured it out, that some of the molecules, sometimes proteins, sometimes not, they can bind to this promoter. And they can bind really heavy. And then we've just seen, maybe I will try to restart this. Now, if the RNAP wants to start transcribing, tries to start expressing the gene, just physically cannot get there. So we turned off the gene. If we have these repressors around, these are molecules, we kill the expression of this gene. So how can we figure this out? How can we understand this mathematically? So here we would have like two states of our system, left and right, and they go from one state to the other. On the right here would be the repressor R being around, but not being bound to the promoter. So that means that expression can happen. On the right-hand side, now it's being bound, so like expression cannot happen. And we can just see these two things as chemical reaction is a chemical reaction network. So the two different chemical reactions, one going from the left to right, one going from right to left. And we would have two different species. So one like this, let's call this D, this binding site, and R, just the amount of repressors that we have. And then we just say, okay, if R and D, they meet, then now the repressor can sit on the promoter and it can block it. So it's a binary reaction. So it's like some constant, let's call it K plus, times D times R. That's what the rate is. And then the other direction, there's always like reversibility going on in chemistry. So if you bind in one direction, you can also unbind again. And this would just be like proportional to like the number of occupied slots. So D-oc would be the occupied slots, and D without a subscript would be the free ones. And then we can, of course, like let's first pick like an ODE model. We can write this down. So the derivative of like the change in deoccupied is sometimes we're getting more, sometimes we're getting less. Less we get whenever it gets unbound. So minus K minus deoc. And we do get more of them, more occupied D slots whenever D and R meet. That's D times R times this constant. And now we're doing something where we're separating time scales. So we're zooming out a bit. We don't want to know exactly how the trajectory goes, like how exactly D gets being occupied and not occupied. But we just want to look over a large time window. Like what percentage of the time do we expect this thing to be occupied? So separation of timescale means going to the steady state, to the limit. In the limit, nothing changes. So we're setting the derivative to 0. That's the first thing that you can look at. If you have an ordinary differential equation, you can look at the steady states. And if you're doing some algebra on this, we figure out that kind of D, so the free sites divided by D tot, so the total amount of sites that are available, so either occupied or unoccupied, is some function of R. And kind of just a sanity check. So if R is 0, if we have no repressors at all, then this is 1. So kind of the proportion of free promoters is 100%. If R is infinity, then it's 0. It kind of goes down with R. So it does make sense in some sense. I was also maybe, talk about maybe this slide and then maybe we can go into the break. It turns out that, of course, this repression, some of these mechanisms are known, so they're usable by us. But in the first place, they're just used by nature. And if we look at our bacteria, our favorite bacteria, E. coli, there's a map of operons, so things that can be regulated, and interactions between them. And like if you count, maybe the count is off by a few, but there's not a complete consensus, but let's say it's 424 operons, so things that can be regulated, can be turned on or off, and 519 interactions. And it turns out that 40 of them are auto-regulations. So kind of regulating yourself. A feedback loop, direct feedback loop on yourself. And in some sense, you can argue that this is overrepresented. Because you would assume, like if everything was uniform, which of course it isn't, but if everything is uniform, you would assume that the kind of number of interactions by number of operons, of sites that can be controlled, is kind of the expected value of kind of auto-regulations. And here, of course, 40 is much bigger than the 1 point whatever that you would get. And it turns out that most of them are negative auto-regulations. And yeah, maybe just say 2 words about auto-regulation. First negative and then positive, and then let's go into the break. So negative auto-regulation, it's weird why you would have that. So you're expressing a gene and the gene pretty much directly turns yourself off. It turns out that this has an accelerating effect on the system. You can work out the math. I skipped it here, but it will be on the slides. And you can also see that experimentally. So here, the authors did two different things. So first of all, they did the math, but also they did the experiments. And they had one kind of system that's just expressing a gene, a regulated gene, but still a inducible gene with autoregulation. And again, I have to underline this. It's negative autoregulation. So the more you express, the more you press yourself. It turns out that the whole system is quicker if you have negative autoregulation. It's weird because you do get you would expect to get less output, and in some sense that's true, but you get there faster on a relative time scale. Maybe just yes. Yes, you just have one gene, and the gene turns itself down. And it speeds up in a relative sense. So it gets faster to 90%. The 90% might be lower, but relatively speaking, it's faster. You can work out the math. It's actually kind of nice to do that. Yeah. So this kind of explain negative autoregulation. Positive autoregulation also has some sense, like makes sense. And more so the positive autoregulation means now the more you are produced, the more even more you produce. So the rate goes up now, the more you have. What that does is a bistable system. So either you have zero, or kind of as soon as you hit the threshold, then you're being pushed to a higher state. And with this kind of, as soon as you have a bistable system, of course, that kind of can remind you of a memory element. And that's what people actually have done is like they implemented so this is with two different genes, a toggle switch. So now we have two different genes, X and Y, and X is down-regulating Y and Y is down-regulating X. So more or less what it does is like there can be only one. If one won, the other one is down-regulated. It's kind of killed off. If the other one is up, the first one needs to go down. And then you have like a memory element now. 1 bit, but still. Yeah, I think that's a good place to go into a break, and then afterwards we will talk about populations. Do you guys have any questions for this first part? So this was only local computation going on, and then we look at distributed. Oh yeah, yeah. I should. So we learned that we can program bacteria. We can put new DNA inside of it. And now the question is like, what can we put on these genes? What's available to us? And kind of what I showed you is like there is Boolean regulation that you can do. You can turn off a gene. You can also turn on a gene. You can implement Boolean gates. And we've seen with a bistable element, with positive autoregulation, that you can implement a memory. Yes, yes. Um, it's impossible and possible at the same time. So it's hard to exactly predict what's going to happen. But sometimes you're seeing trends. Um, oh yeah, of course, you just do the experiments. I'm kidding. But yeah. Yes, yes. So you can do a few different things. So you can like have your system experimentally, of course, is the ultimate goal. Like the ultimate arbiter is experimental. But you can also like simulate it in a more detailed model where you take into account more. Maybe you cannot reason about it mathematically, but you can simulate it still. Yeah.
how can I so challenge is all concurrently so that like I could get some inspiration from biology even
Yeah.
like the take
Yeah.
is there anything to gain from can I like are there any concurrency things which happen in this chemical experiment?
Yes. Yeah, so everything here happens concurrently. It's not like it's much less quote unquote discrete as we usually would want it to be. So kind of it's much more less discrete and more probabilistic at the same time. So this is something that we don't have at all. Like if you program your, I don't know, your lock or something, or your garbage collector, you wouldn't have to deal with it just failing 10% of the time. So in that sense, kind of, I don't think you will get. Maybe you can get an inspiration, but it's not exactly what you can get out of this mod looking at these models. I think it's more or less the other way around. So kind of looking at these models can be really useful because then you can maybe argue or reason about the systems that are of interest. As a byproduct, of course, many people do, for example, population protocols and kind of it's some theoretical interest there because sometimes the techniques are transferable, but kind of the results being transferable like to a computer system, I don't think that's exactly what's happening. If that were a question.
Direct activity going on text because I've heard of so like ant colonies
Yeah.
like
Yeah.
so
Exactly.
they can have some balance there but
Yeah. Yeah.
solving some
Yeah. Yeah. That, for me, that's a different way. So actually, I came into this business because I looked at bird flocking, and then I wanted to really know how that happens, and I really wanted to look at birds, and I like decided for myself it's too complicated. Individual are too complicated, and then I looked at what is the simplest way, like what is the simplest living thing? It's bacteria and it's E. coli, and then I tried to understand these. But for me, it's bio inspiration here. I'm sure there is some. But the work that I'm presenting here is not the goal is not that. That's a very good question. I don't have the numbers, but there are some back of the envelope calculations that show that even for like Boolean computation, they can be much more energy efficient than electronic computers. I don't have the numbers in mind. Yeah. Even taking into account that, of course, like what that doesn't take, like more or less, like it compares the energy that's stored in the sugar cube that you can put in on the things and the electrical energy. What it doesn't take into account is what you need to get there. And how you do the readout. Like there would be, of course, like these curves that I'm showing about fluorescence readouts. Of course, there are computers. Yes. Yes, I didn't do the calculations myself, but that's what I read. Yes. Yeah. Um, okay, so we've been talking about how to program cells, bacteria, and now let's try to do some distributed computing with that. So the first question is like, how should we model? Like we've been modeling what's going on inside of a cell with chemical reaction networks, with CRNs. And a priori, like if you think of like now a collection of bacteria, you would maybe think that now you have to keep track of all the different concentrations of all the different molecules in every single cell. And there's so many cells. Like in 1 milliliter, just to give you a rough idea, there would be 10 to the power of 9 bacterial cells, like in a typical colony that you would see, which is of course unfeasible. Like maybe you can do some proofs about this, but for simulation, for example, it's totally unfeasible. So the thing that we are currently trying, like the approach that we're currently doing, and it's implicit or explicit also with other people, is like to reuse the chemical reaction network formalism, but now on a population level. So now our A's and B's will no longer be molecules, but whole bacteria. And they might have a state change, but they might have an internal state, but we'll come to that in a second. So here just three different chemical reaction networks for very simple systems. So it's only population growth. So just a growing colony of bacteria. So the first one might be the most simple one. So just C goes to 2C. And here this is a log plot. So it gives you a straight line. And of course, that's not what you observe. You don't observe unlimited growth a priori. So what people sometimes do is like now you're introducing like C would be our cells. And now they're introducing like R, which is a resource. R is now not a cell, but it's a collection of molecules that lives outside and that you can maybe import and that you can use to duplicate. So now kind of you C and R, they have to meet. Like physically, you have to take up these molecules. You can think of them as glucose, as sugar, or amino acids, or something else. And then once you take them up, now you can duplicate. So these would be two different formulations of kind of now population level models, CRN models for bacteria. And then they can get cute with that as well. So like here, for example, like on the right, I just put you that as something that you can also do just based on this very simple model. And this actually gets closer to what you see in experimental data. Like often you see like a two-phase growth, and you can have that with two different resources, one being faster than the other, but taken up faster, like running out faster than the other, and so on. Yeah. 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 Yes, there is an abstraction here going on. So C and R is not one molecule. Like what happens is like C needs to take up over time more and more molecules that it can extract energy out of. And at some point it will duplicate. So what happens is it's like this would be a very non-atomic process. Like you take up things of like here we are abstracting to like a one thing point. So there's a separation of time scales that we're doing here. Yeah. You can look at things that take up things over time, but it gets more and more complicated. Yeah. Okay. So I just want to present this. This has like this is an experimental and slash theoretical work that we published last year, and what we did there, and just to introduce like what we how we can work with population level models in real like model real biological systems. So what we did here is like just a communication system, and we implemented a communication system from sender cells to receiver cells based on viruses. Viruses are, everybody knows what a virus is. It's genetic information inside of a box. Since COVID, we do know that. And there are, of course, viruses, already saying that at some point we discussed this, there are also viruses that attack bacteria. And we can just take one of them, and we can also modify use our DNA that we introduce into the bacterium, that it gets introduced, gets packaged up by the virus, and then it's our DNA floating from one cell to the other. And if you do that, and a few other things, is that what you would see is things like this. This is a part of the chemical reaction network that we constructed for this work. And what you would see, so here, for example, it's just growth again. So C and R, again, it was R1 and R2. Both of them happen here. And now we have C, but every C has a state attached to it. So here we say like C, if can equals to false, can is like kanamycin, it's an antibiotic. And most of the time you are selecting bacteria based on antibiotic resistance. So kind of you're selecting a subpopulation by killing off all the others, and the ones that are resistant, they live. And so like for example, like that, yeah, what I'm trying to say is like you have all these internal states. So for example, like a newborn cell C here on the right hand side would have a low infectability for the virus. And if it grows over time, like if it gets older and older, the infectability gets higher and higher, more or less because it builds up more of the receptors that the virus can attach to. With this, I just wanted to say that now we do have at least discrete states of our system. So maybe we don't have to keep track of all the continuous state, like exact concentrations of all the molecules in all the bacteria, but we can do sometimes abstractions. So one would mean like infectability equal to low would mean a low amount under a given threshold amount of receptors. And then above would be no, you're above the threshold. So you're doing like a discrete state abstraction here, and yeah, this writing down these things bugged us so much that we invented kind of a programming language for that. That's just more or less based on like Cartesian products of state spaces, and it's just a multiplication that we use here. This is Python code. Um, like you can have like you could be low infectable and high infectable, and at the same time, maybe another direction might be your age, like young and old, and all these things can happen. Not maybe in all combinations, but at least you might want to have all combinations available to you. Okay, so now I want to turn to like a much simpler system where we don't have so many internal states, but where we can prove things about this. Like we will have a mathematical proof in a few minutes about bacterial systems. And yeah, just give the usual spiel about distributed systems. Of course, like we do know now that we can program bacteria, but if you put too much of your own DNA inside of a bacterium, they will just be so busy doing your work that it will die. Or maybe they will select away from your program. So only the ones that don't have your program will live because they outpace the others. So of course, like the natural approach is to distribute things now. So like let's say you have some computation, you cannot put everything into one cell. So now you're putting it into multiple cells or cell types rather. And that is nice, of course, because now every single cell type has, or every single cell has less burden on it. But now you have to communicate in some way. And yeah, of course, the communication is also tends to be very, very noisy. We will talk about the specific mechanism, how we implemented the communication. So we can do it with viruses. That's not how we thought about this here in this theoretical paper, but there's another mechanism, another biological mechanism that we used. Okay, but just like as motivation of the problem that we want to solve here. We do want to solve, let's call it signal amplification. And why do you need that? So let's pick here the first example here. So here, let's say we have three different Boolean gates or parts of a circuit, A, B, and C, and maybe they're in sequence. If you put all of them inside of a cell, it's very likely that kind of every single one of them, every single component has a certain probability of failing. That's usually what you see, kind of that's where stochasticity also comes from, or that's a corollary of your stochasticity, that each one of them has a certain probability of failing. So here on the right-hand side, I just put like a theoretical simulation where we said every single one of these gates, A, B, or C, has a certain probability of failing, I think 10%. And then you see kind of on the right, top right here, like the after each stage A, B, or C, kind of you go down with the fraction of correct cells. Maybe the first one goes up to 90%, but then the second one goes to 81% and so on. And now, of course, you can distribute, but this doesn't change this problem, right? Because, like, let's say A now communicates something to B, but there's a certain percentage of A that's just wrong output. And then B collects these things and produces its own output, but some of its input were faulty. So kind of it amplifies this error from the start. And then there's of course this approach that of course is the one taken by electronic circuits, which is just to put like an op amp in between, like a signal amplification. So kind of even if you're not 100% correct or you're not at 100% of your voltage, of your target voltage, the transistor will pull you up. The op amp will pull you up. And now the question is, of course, like, and then of course you get what you see here on the bottom right, kind of that A, B, or C kind of the because in between more or less you're pushing everything to being 100% or close to 100% correct. You get this very nice outputs even at after stage C, even stage D, E, and whatever. And then the question is like, can we implement this differential amplifier or sorry, this signal amplifier in between two gates? The answer will be yes, but first let's talk about like how we want to communicate. So viruses is one possibility. Quorum sensing that I've already told you about at the beginning is another possibility. There's another one that's based on direct contact of cells. It's called conjugation. And when two bacteria come close to each other, they can open up a channel, like a physical channel in between, and one bacterium can push some of its DNA, copy it actually, to the other. That's how much of the antibiotic resistance gets passed around among bacteria. Like they have these pieces of DNA with a resistance gene on it. They just give it to their friends, and that's why we are in the mess that we're in. Based on with respect to antibiotic resistance. Why did we pick that? Because it's mathematically simpler. Direct contact would mean something that we can write a rule that's here on the bottom left. A plus B does something. It's not like A producing a virus and the virus needs to go to B and then B and the virus does something else. Just A and B. Whenever they meet, they do something. And this is actually the whole protocol that I want to talk about. It's the protocol proper is actually just the last line. A plus B goes to nothing. So what it would mean is kind of A and B exchanging genetic information so that both of them will die. We already know that they can't do that, right? So they can have a trigger that they kill themselves. The first two lines, A goes to 2A, B goes to 2B, there's no choice. This is not us. Like just when you talk about bacteria, that's what they do. Right, and then I I put this here as a quote unquote exercise, but like as soon as we have this chemical reaction network, of course we can write down the ODEs. I would rather talk about the stochastic semantics for it because more or less the deterministic ODE kinetics is not very interesting. If you write down kind of the stochastic semantics here, is you have three different possible successor states in each state. So a state is, I remind you, count of A, comma, count of B. And you have three different events that can happen. So duplication of A, so A goes up, duplication of B, B goes up, or A and B meeting, A and B both going down. They meet and they annihilate each other. And this is why we call it this pairwise annihilation protocol. And and each one of them has a certain probability of happening. So here, this is the jump chain, the discrete time jump chain, not the continuous time chain. More or less, what you're taking here is kind of you on the numerator you put the rate, and again the rate is rate constant times the product of the reactants. So for the first one, it's just A, so beta times A, and then you divide by the sum of all the possible reactions in the system. There are only three, so it's like beta A plus beta B plus alpha AB. Okay, and then you can of course like simulate this. More or less, what happens is here, like sometimes like we have A and B, and sometimes A gets duplicate, and sometimes B gets duplicate, and if A and B meet, they annihilate each other, and at some point you can prove that with probability one, like one of them will die out. But this is of course a stochastic process, so we haven't asked thought about exactly what question to ask, but this is just to undermine that, to underline that this is like a stochastic process. So here, for example, like there are some stochastic runs with a majority of B's, and delta is the initial delta between A's and B's. And here, for example, like with A and B 1,000, so it kind of A would have 997, and B would be 1003, and here B would win, and maybe with the 30, like B might also win, but it's a bit more more stochastic here. Yeah, just I already said that at the beginning, like that, or in the first part, that there is an analogy between population protocols and CRNs, and a very similar looking protocol has been analyzed. One of the early population protocol work actually has looked at this at more or less like at when A and B meet, they cancel each other more or less. Um, this has been used to solve a certain problem, which is the majority problem. So you have A's and B's, and you want to know who's in the majority initially. And of course, they cannot really like kill one of them. So they have to change one of them to a blank state. And then they do also have a duplication actually. So when a blank state meets like something that's not blank, like they rise them, like the non-blank one gets duplicated into non-blank state gets duplicated into the other. Of course, like the kinetics are very different, but the idea is definitely there. There's also related work in terms of control theory inside of bacteria. So this is with molecules. This is more or less what they wanted to implement is to implement the difference of two different molecules, molecular species, to do like a control loop. They wanted to do the error, and for this you need like a subtraction. And yeah, if you build that this was 7 years ago, but like it's still like if you implement something like a minus inside with molecules inside of a cell, or this wasn't even inside of a cell with molecules, you easily in nature, because also of course they built on top of that like a control loop using that. But the mechanism was that there was one molecule and a second molecule, and whenever they form the complex, whenever they hit and they form the complex, they inactivated each other. So kind of they were still around physically, but they were inactive. They couldn't interact with anything. Okay, let's go back to ours. So simplified, just A can duplicate, B can duplicate, and whenever they meet, nothing happens. So what we could prove, and where we talk about in the end why that's useful for signal amplification and for Boolean computation in general. But what we show is, or what you can show is that kind of if you're starting out with an initial population n and some initial gap, so that's the difference between initial counts. If the gap satisfies something, and we'll talk about what, then our protocol reaches majority consensus with high probability. So what it means is kind of, I mentioned that one of them will die out, and it will be very likely the minority that will die out. And we see here that kind of the delta that you need, like this really like A minus B, is sublinear. It's more or less square root n. So you can refine the thing, but in the theorem that I will kind of try to prove to you, it's square root n. So that's not even, you can think of it, you don't even need 51%. 51% would be a linear difference that's required. It's even sublinear. And then you can look at the time until consensus, or until one of them dies out. So this heat map, maybe let's only look at the right one. So here it's a heat map. So if it gets more and more yellow, brighter, then it takes longer time, and if it gets darker, then it's less time. So more or less what you see is like if A and B are close together, that's when it's difficult, of course, and that's where you need a long time to stabilize, quote unquote. If it's clear who's in the majority, then it's pretty quickly you figure it out. Yeah, and here this is the metric that I was presenting in the theorem. So that's the probability of majority consensus. Also, specifically here is the probability of species A winning, meaning being the only species that's left. And so this is the probability on the y-axis. On the x-axis, it's the initial delta. So A0 minus B0. But what you can see is like if A is equal to B, so if the difference is 0, then you have 50%. It's just because it's because of symmetry. We assume that both of them have the same birth rate. And then kind of you can see that kind of the further you go to the right, let's say, the higher the probability that A wins. So now A is A minus B is positive initially. So if A is in the majority, so it's good. That's what now we want to decide that A is in the majority. It's never 100%, but it gets close. And what's interesting here is that bear in mind that on the x-axis, it's just a difference, like the counts. It's not like ratios or something. And here we have like in the different colors, we have different initial counts. So the relative initial delta gets smaller and smaller and smaller. And you can kind of see the probability curve degrading a bit, but not too much actually. So here we are jumping one order of magnitude at each time, and it goes down a little bit. So how to reason about these things? Now we are in continuous time Markov chain land. So kind of there are all the tools that we know and love and that at our disposal. And so yeah, the two things that or maybe I won't do the time. Maybe just let's talk about the probability of majority consensus. So the way we initially analyzed it, we refined it this afterwards, but in a very simple way, you can think of your system in the following way, or you can couple your system with a much simpler system. Coupling in the sense that kind of if one of them reaches a favorable state, and the other one also reaches a favorable state. And you can think of this as an urn model. So here, more or less, we're only looking at the birth component of the system and not about the annihilation part of the system. So more or less, why? Because the annihilation part, A and B meeting, doesn't change the difference. But if A duplicates or B duplicates, that changes the difference. And here we like the coupling that we did was based on the difference. So if you're only thinking about the births, so what you can think of, like you have some orange balls and green balls, and they're in an urn, you cannot know what is what, but you grab into it, you get one of them, you look at it, and whatever the color is, you take a second one and put it back. More or less, that means like you uniformly sampling from that, and whatever color you get, you put one more of that. You're amplifying this. And this is exactly what happens if you're forgetting or if you're omitting the annihilation reaction, so A and B killing each other. If just you have A duplicating and B duplicating and they do it at the same rate, that's exactly what you're getting. Like because the probability then will just be beta times A over beta A plus beta B, which is just A over A plus B. Just the proportional count. Right. And once you have that, you can figure out that kind of the probability that the minority wins is proportional to the probability that you at some point you get the equal count, A being B. Initially, maybe you wouldn't have that. Initially, you were separated, but there's some probability that you're getting exactly the same count. And then let's say A is in the majority at the beginning, so A should win. And then let's look at the error probability that B wins. So this, of course, we want to show that this is small. And then we can just apply the law of total probability and decompose it based on this event that there exists a t so that AT is equal to BT, so that they're exactly the same count. And so what you can notice here, so of course, like you do it for existence and then for the opposite, so for RT, they are different. If they're always different and A was initially in the majority, then actually B cannot win. Right? At some point, the majority needs to flip. We need to have the same. So the second term actually, it's zero. That doesn't exist. And then there's also like because of symmetry, once and the strong Markov property, as soon as you are in the state, if you study out your system in a symmetric state A equal to B, the first thing is just 1/2, just based on symmetry. You can restart your system, you can regenerate your system in the state that exists here. That's this is a stopping time. And so by the strong Markov property, you can just start your system there, then it's 50-50 who wins. So the whole thing is just like the probability of B winning is proportional, like it's a factor of 1/2, but the probability of A and B ever being equal. So this was for A and B. Now we can go to the earn process, and the probability that at some point we get in the earn process the same amount of orange and green is also related to another thing that we can measure actually. We will see in a second why we pick that, but we can look at the probability that kind of the limit of the relative counts of orange is at most 1/2. You can show that with probability 1 the limit exists. And yeah, we see kind of just to say that right now it's like kind of it turns out that this limit not only exists with probability 1, but it's distributed by a very well-known probability distribution. It's a beta distribution. Right, so as soon as you're talking about these things, you can just look up the PDF on Wikipedia and you can reason about that. And so here we can do the same trick, for example. So here now we're looking at the probability that the limit average count is smaller than 1/2. And by the same token, by the same argument, we can like apply the law of total probability for the existence of t so that orange is equal to t at some point. The second term I didn't even write because it's again 0. At some point it would need to flip. But also like the first term here, it's again equal to 1/2, because now if we're regenerating our system in the state that in the stopping time that the count of orange is equal to the count of green, then again it's 50-50. And the coupling, kind of the way that we couple our birth and birth system to the earn process guarantees that if we're hitting A equal to B at some point, then we also hitting orange equal to green at some point, because I didn't present it here, but it's based on the difference between the two of them. So if one difference goes to 0, then the other difference also needs to go to 0. And so if the second one is improbable, then the first one also needs to be improbable, because there's an implication between the two. And then we have the bottom line here, so that the probability of B winning, so kind of the error that the initial minority would still be able to flip the majority and win, is kind of the first thing we already did on the last slide, then the implication. And now the last one here is related to what's called I 1/2 of A0 and B0. This is the CDF, the cumulative distribution function of the beta distribution. It's just something that's like there are books about earn processes, and one of the first results is that if you have these earns where you put in the second one, whatever you picked, then you get the in the limit you get the beta distribution. Just smart picture here. So here it's the PDF for different values of initial orange and green, and the initial orange and green will be initial A's and initial B's. And what we're interested in is like the integral. That's what we want to know. And here we see kind of if the count of orange and green goes up, or the ratio, or the difference between orange and green goes up, kind of this gets smaller and smaller and smaller. It gets more heavy on the right side. So the left side will be smaller and smaller. Right. So that means that kind of if we can reason about this what's called the incomplete regularized beta function, which is, you can look it up on Wikipedia. It's and the analysis behind it is not too bad. Like you can do it. It turns out that now if you put in kind of our guarantees that we have on A0 and B0, which is again that we gave names to the sum and the difference. The sum was n and the difference was delta. So the initial gap. So here kind of more or less we're starting them out in the worst case. So n plus delta for the majority, n plus delta over 2 for the majority, and n minus delta over 2 for the minority. So if you sum them up, you will get up to n. If you do the difference between, you get exactly delta. Yeah, you can do the math. So in the last equality sign here, it's kind of some analysis, not too bad. You get that this is like an e power minus omega of delta square over n. And this delta square is where kind of the square root in the gap comes from, because now if we're putting in like delta equal to what we proposed in the theorem, so square root n log n, then of course you get like the square of this is n log n over n, so the n disappears, and then you get e power minus log n, so 1 over a polynomial, which is kind of what people call with high probability. So the error is bounded by 1 over a polynomial. Okay. Right. So this kind of proves that kind of that the gap that you need is only square root of n. It's not like linear as you might imagine, but it's just square root of n, which is good news. And then in further work we're still doing that actually, but we're trying to make the models more and more realistic in some sense. So first of all, something that we couldn't do with our EARN processes or other proofs based on EARN processes is we couldn't, and I left that out actually, we didn't have cell death, individual cell death, that is. So mostly like bacteria, they always duplicate, but they also die with some rate. It's not too high, but it always exists. So here, the kind of on the right-hand side now, we're putting in, like as for any species, for any bacterial species, X duplicates, but it can also die with some rate. So this is one additional like realism that we introduced. And then also, I didn't tell you the full truth when I told you about this conjugation mechanism. So whenever A and B meet, they can open up the channel, but it's a unidirectional channel. It goes one way or the other, not both at the same time. They can do it in 2 different steps. They can do it, but there's always one initiator and one receptor, and one receiver. So actually, this A plus B going to 0, this is not exactly how you can implement things. What you can implement is A plus, or what we can implement more easily is A plus B going to just A. What that means is kind of, you can imagine that if A and B, each of them have some genetic information that if they're combined together in one bacterium, this bacterium will kill itself. More or less, what you can think of is like one of them upregulates the killing gene in the other. So maybe B will be the killing gene, and A will be the upregulation of that. And we already know that kind of we can implement like switching genes on. Right? So A and B, they will happily live. But then if let's say in the first direction here, so A plus B goes to A, here A would transmit its part of this pair to B, And now this new B cell would have both parts, so it will die. And this can happen in the other direction. So not atomically, it cannot happen atomically in both directions, but it can happen also. It's just another path that can be taken. So A plus B might be that B transmits into A, and then A will have both parts, and thus it will die. And there's a separation between these two models. So kind of the old model where A and B directly die, both of them die at the same time. Might talk a bit about the proof techniques, but we could prove that kind of that the square root n kind of it was a nice analysis based on urns, and it was kind of relatively easy to think about. But actually, it's like more or less you're around polylog. You only need a polylogarithmic difference for the majority to be correctly identified, which sounds crazy for any biological system because you would expect more noise out of it. For me, this is also like just a sign that our model is not the correct one. When biological systems can detect a logarithm of the difference, sorry, a logarithmic distance, a difference that's too precise. It's nice math, but it's too precise. The other model, where now it kind of only one of them can die at the same time. Both is possible, but in each event, only one of them can die. Then actually we showed that, or you can prove that kind of the square root thing, the square root area is where where the reality lies. So we do have like a lower bound and an upper bound. The upper bound here it's proved with other techniques because now the two differences between that, like first of all, we have the death reactions, so it kind of rules out the urn approach, and we have the non-symmetric killing annihilation reaction. The lower bound, maybe just to say a few words about this. So the lower bound is more or less just based on um, you would have just A's and B's, and you can have like A going up or B sorry, A going down or B going down based more or less on a roughly normalized roughly normal distribution. So kind of it's an inverse central limit theorem that we sorry, an inverse with the central limit theorem that we're using here. The upper bound is based on a Summa's inequality, but so you need to do some more work. But what I'm trying to say here is like there is some area, and I don't know exactly where it stops. We haven't figured that out, where you still can do proofs. Maybe they get harder, but you can still do proofs being realistic. At some point, it stops. At some point, you will not be able to do things anymore, and maybe you can do simulations at some point. Maybe you can only do experiments. But it's not clear where exactly it stops. And here we could go on, for example. Maybe just one lower bound that's kind of I hope easy to see. So I want to convince you that okay, so maybe you buy that kind of if we're implementing this annihilation reaction, A's and B's killing each other, maybe we get to majority consensus. I want to prove to you, and I think I can give you the complete proof that if we don't have that, so if there's no interaction between A and B directly, there's indirectly, sorry, there's no interaction between them at all. So X, like A can give 2A, B can give 2B, A can die on its own and B can die on its own. Then kind of the probability, the error probability, so the probability that B wins is just the relative initial count of B, which is kind of really bad. So like if you're close to 50-50, like if you have 51%, then you have like a 51% majority, then you have a 49% probability of doing the wrong thing. And it's just based on the following. So we can, let's say initially you have A's and you have B's, and each one of them can give rise to a lineage. So having some daughter cells. Let's say you keep track of them. So each one of them, either maybe they die out, the lineage can be finite or can be infinite. But if you have if you Sorry, I forget uh, one important detail. So sometimes you can show that at some point on a very long time scale, you do die out. Maybe just based on like happenstance that there are too many death events that lead you to zero. If you have that, then there will be a last lineage that stays alive. Right? All the lineages are equally distributed, identically distributed. The IID, so one of them is the last one standing. They're all identical because we assume here that they have identical rates. Beta is the same for both of them. Delta is the same for both of them. So just one of them will stay in the end. And each one of them has an equal chance of being that last one. So it's just the initial count. So the initial proportion that is the probability of it being the last one standing. If you're 50-50, then it's 50-50. If you have 30%, like let's say you have 10, and you have 3 that are A and 7 that are B, that is a 30% chance that that's A and 70% that it's B. Right. So I hope with this I convinced you that if there's no interaction, or at least no direct competition, then the probability of B winning is just its initial proportion. But what it means is kind of for our thing, we wanted to have like with high probability, we wanted to have the majority winning. And for this whole thing, like B0 over A0 plus B0 to be 1 over n, that would mean that B0 would need to be constant. That in particular means that kind of the initial gap now needs to be linear. So kind of we went from this super precise model where even a logarithmic change can be detected. Seems kind of unrealistic. A more realistic one where you have the interactions, but it's a square root gap that you can detect. To if you don't do anything at all, you would expect that you're bad, and you are very bad. You need to have like a linear initial gap. Okay. So with this, I hope I convinced you that we can, in principle, implement these signal amplifiers. Why is that a simple signal amplifier? Because like, let's say your signal, so like let's A's and B's are the output of your system. One of them will be the correct one, either A or B, but like let's say you're more than 50%, you have a higher than 50% likelihood of having the correct answer, which is, if it's a binary answer, it's good to be there. Otherwise, you wouldn't be able to do anything. But as soon as you have that, then you're over 50%. So that means that initially you would be in the majority. You would have a majority of correct answers. And then kind of the signal amplification would mean that you can even drop down the wrong answers to 0. So the initial majority really winning and the others really dying out. So how can we now use that? So we did some theoretical analysis on that as well. So now we assume that kind of on top of you being able to do signal amplification, we also want to do like Boolean circuit. So let's say here, like in a few different cells with different parts of the Boolean circuit, and they can communicate their answers. And then you can, what you would need for the signal amplification to work, you would need to have like a dual rail encoding of your output signal. Dual rail encoding usually means that, like, if you want to communicate a one, like more or less it's a one hot encoding of your output. So like you have one line for 0, one line for 1. And if you want to communicate a 0, so then you have to raise up the 0 line and leave the 1 line down. And if you want to signal a 1, then you have to keep the 0 line down and raise the 1 line up. This is often considered to be more robust even in electrical circuits. And here it's kind of necessary because we need to have 2 different species that can interact for our signal amplification algorithm. Okay, and let's say now the A0 and A1, so this dual rail encoding is like the output of a gate. And we introduced like what it means to be n delta correct. Yeah, more or less what it means is that that both of them together have a non-trivial size bigger than n, and the delta is bounded. So the gap is at least delta. And now we can kind of build up these noise-tolerant Boolean gates where, first of all, we have everything is dual rail encoded. So there's only two inputs. There's A, B, and the output is Y. But it's dual rail, so you have 4 input lines and 2 output lines. And this triangle here on the right is kind of our signal amplification. And if you put these things together, then kind of you would have now like A's and B's interacting, and when they do, they can also output a Y species, which is based on the Boolean combination of these things. And this is a bit even messier than the other analysis, but you can show that kind of if A's and B's are initially n delta correct, so then with high probability, your output will also be like n delta correct. And kind of this is I'm not going to go into the proof here, but more or less the difficulty that you have here is the kind of you have the noise at the input, so that's a given in the initial state, but you also have the application of kind of the wrong inputs and the correct inputs. And with this, kind of you have two different noise sources at the output. You have the application of correct or wrong outputs, which is of course a problem. So kind of you have to be a bit more careful, but you can do it in the end. So putting these things together would mean that you do have kind of these Boolean gates. So here we only did it for NAND, but that kind of do have the signal amplification attached to them, which is very close to kind of like a Boolean gate, a logical gate in your electrical circuit. Right. So just let's look at some simulation results. So here, there's just again like in our model, like we analyze it, of course, analytically, mathematically, but of course we did it in a asymptotic fashion. Like we talked about big O, like omega of n, omega of square root n log n. All these things are asymptotic. Why? Because that's what we're used to. This is much easier than doing the real thing. So to get like the numerical values down, we did simulations, and it turns out that kind of it seems to work out fine. So here we assumed like an input error of 10%, and we do have like the outputs here. Let's pick the one where the green line points to. The correct output is the blue one, and there is a separation between the two. And this could go now into like there's a gap, and this gap is not just like additive. It's a log plot, so this would be a multiplicative gap even. So the other line, the incorrect output is much lower than the correct one, and this can then go in to kind of the input of the next gate. So we can, of course, NAND gates are universal, so kind of you can put them together and now expect that after each one of them, you do have a signal amplification that goes you up to reasonable value. And here we showed that this is possible. Oh, yes, this is ours. It's already kind of quick for bacterial computations. Yeah. Yeah. So here everything, as soon as we're talking about like as soon as if we put the application in our system, that means we're on the time scale, at least approximate time scale of the applications. The application is around 20 to 40 minutes. So being 3 hours is already kind of quick, actually. This is simulation. Yes. We have not implemented this. So we are doing the implementation not of the whole Boolean gate, but of the signal amplification. It's slower than this. Like you yeah. Yes. So the most one is the one where kind of if A and B meet, then either A dies or B dies. Both of them dying is 100% unrealistic. And we knew that before because like conjugation is a unidirectional form of communication, but it also sounds crazy that you can detect like a uh, logarithmic difference between two signals. That's too good to be true. Yeah, exactly. That's where it is. Yeah. Okay. Yeah, with this, I want to conclude. I gave some open questions and I want to show you some pictures of our lab afterwards. So kind of get a feeling for kind of how these things look like if you implement them. Right. So just there is a a motivation for why to look at like synthetic biological systems. Uh, for me, like bioproduction is fine. In vivo, so meaning like in the body of animals, us or others, diagnosis or therapy sounds very exciting. Of course, there are many challenges. And many of the challenges for us at least are like finding the appropriate models, the correct ones. Like the CRNs, it's as a class of models is fine, but then you have to think about what are the directions exactly, and so on. Yeah, and then kind of I hope what I showed you is like makes you tend to believe that some of the aspects of these systems you can analyze, understand, and prove mathematically. Um, even distributed like bacterial algorithms. Everything is not feasible to prove. And simulation is fine. Other approaches are fine. Yeah, and then just to conclude, maybe about very specifically like about the big open questions as I see them, at least. Kind of what would need to be done to kind of push this endeavor forward is so we've been talking a bit about like what are the algorithmic primitives, like what you can do locally exactly. So some of the mechanisms are known. Some of them, I guess, are completely unknown. But even about the things that are known, I think there is an effort to be made, like to catalog them, to know kind of what is possible. Um, because you cannot put everything together at the same time. There are limits in terms of size, in terms of exactly what you can implement. Composability is a big issue, actually. Like things cross-reacting, like you're implementing one thing and a second one in the same thing, but then if you put both of them inside, maybe it breaks because there's a cross reaction that you didn't think of or didn't put in your model or something. Um, then there's also this. As soon as you go to the population level, we had the discrete state abstraction that we did, where we said, okay, every bacterium now like is the state of every bacterium is discrete. It's not like a continuous collection of continuous variables, which is the concentration for each of the molecules of interest. And like this has to have its limits. And there needs to be like I think an exploration of exactly when it is valid to do that and when it breaks down. Also, I've already been saying that at some point, already said that at some point, the validity and limits of asymptotic analysis. Actually, it's unclear. Of course, it is unclear in algorithmics, like in classical algorithmics. Here, it's also unclear. Like an improvement of like 50% might be like a breakthrough result in bacterial systems. 50% in some areas also in classical algorithmics might be, but we're much more used to just to writing square root n, O of n, and so on. And like on the limit of in the let's say in the range of parameter values that we see um, in bacterial systems, like it would need to I think need to be thought about like what exactly it shows, like when we show that something is square root n or something. Yeah, and of course, then as soon as you have like the algorithmic primitives down, like you have like and communication systems, uh, then you can think about like a proper theory of what is algorithmically implementable, what is impossible to implement a priori, where you don't even need to try. This, of course, you can only do once you do have like a tool case of things that you're happy with, that you think is covers many of the things that you might be able to implement. Okay, I just want to leave you with some pictures of how these things look like. This is our lab. So here we're growing bacteria. You see a few things here. One of the more funny things is kind of this glass, the tables with the glass in front of them. That's very funny. So but whenever you manipulate like bacteria, you want to like you open up the glass a bit, and then you put the bacteria in, and then you manipulate it there. And it's to protect. It's meant for protection, but not for you. Like there's an airflow going, but it pushes against you. It's that you don't introduce anything to the bacteria. Of course, we only work with non-pathogenic bacteria. If you do work with pathogenic bacteria, you shouldn't do that. You should be protected yourself. We do wear gloves, by the way, but no, yeah, contamination is an issue. Yeah, and kind of more or less it's the things. Like here, you see some incubators. Um, I think you see that the one in the middle here that's died to 37 degrees. Some of the bacteria that love 37 degrees. That's our temperature. On the right hand side, so you see this is actually not bacteria. This is microalgae. We also do some microalgae work. Microalgae are like they're not bacteria, but they're also unicellular organisms. So they're also small in some sense and understandable. And these are actually so yeah. This is how you would kind of have some readout of your bacteria. So here it's a petri dish with each dot that you see here is a bacterial colony. Not individual bacteria, they're much too small, but one colony. And then you can do these things to, for example, to select bacteria that you want. So we've been talking in the beginning about like introducing DNA inside of your bacteria, like DNA that you want them. And then like more or less like I told you, you shock these things and then you put in the bacteria and the DNA at the same time and you hope that something sticks. If you do that, not all of your bacteria will have your DNA inside of them. So how do you filter out the ones that that did take up the DNA? It's very brutal. Like microbiology is sometimes very brutal. Um, you put inside of the DNA that you put inside also an antibiotic resistance gene that the others don't have, and then you put just the antibiotic. All of them will die except yours. That's it. Happens over and over again. And here we did that. So you would you would like have your petri dish and put the antibiotic everywhere, put all the bacteria and the ones that stick here, the ones that live to build up a colony. That's the one that you can pick. Usually you take a toothpick and then you stab them and then you put it inside of new medium. Yeah, this is an example of communicating cells. Again, the readout is kind of boring here. It's just like means that yeah, some bacterium gave something else, like another antibiotic resistance gene to a bacterium, and then we saw that. Yeah, this was based on the phage, sorry, the virus that I've been talking. It's called M13. Yeah, you can do other things. Like we also have you can look at things under the microscope, of course. So these are microalgae. They're not these rod-shaped bacteria that we saw. Tend to be green sometimes, tend to be round. Bacteria, so this is not a very sharp picture, of course. You can get better than this. But these are E. coli here. They're much smaller. I think the resolution is the same on both, or maybe even it's more zoomed in here. Yeah, but the scale is quite a bit different. Of course. Yeah, yeah. So in the first one we did, we did apply with a pump. We did apply a water flow. Here actually we didn't. So it might be maybe maybe there was a slight skew here, but no. So yeah, so these are immotile E. coli. So some of them they do have like a type of propeller. They can move on their own. These ones they don't. So most of the movement is like Brown in motion, like being pushed around by water. Why they do have the tendency to go up, I could not tell you in this video. Maybe just our table was a bit skewed. This is real time. Yes, yes, yes. What's that? Yeah, yes, yes. But this is yes, 100%. The probability is really low. It would take a long time, but of course, this is not the density that we've been targeting earlier. This is a really, really like sparse setting. But 100%, yeah, here would take a long time. So more or less, we did this. We wanted to do this because we wanted to zoom in. We didn't 100% succeed still, but we want to zoom in on kind of the conjugation process itself. And there we don't want to have too many of them happening. So we want to catch them sometimes. And when we do, we want to realize that we're catching them doing the conjugation. That was the setup. Right. Yeah, that's lots of people that work on it. Most of the people, are, of course, like past lab members. But of course, it is kind of a different game if you want to do biology. No, no, no. Biologists, computer scientists, physicists, control theorists. I think most of them are computer scientists, but no, no, by far not all of them. Yeah, we do need this. Yeah, but very much so, very much so. Yeah, yeah. So we kind of we have a few people like doing the math work. We have lots of people actually working on computational models, computational methods also. Like simulation is not only one of them. Actually, one big headache that we have is like we build up these models, we write them down, but then we don't know the constants. So parameterization is a big issue. So it's optimization on time series. Takes up a lot of our time. And then we have some experimentalists also doing the thing. Yeah, yeah. Yes, we need to. Yeah, yeah. So we don't do all of our experiments on our own. I think one of the things that I showed you with the viruses, we didn't do in-house, we collaborated. I'm trained as a computer scientist. Yes, yes. I yeah. I can pipette nowadays, but I didn't. Like 5 years ago, I wasn't able to pipette. But it's not rocket science. You just have to be careful. Yeah, yeah, yeah. I think that's all I wanted to present to you. If you guys have any questions, I'll be happy to answer them. Oh, that's a good question. Yes. Yeah. We're exploring these things, so we haven't done it yet, but this is something that's startup adjacent some of the time.
You think that eventually there might be some kind of a product?
Uh,
Like,
yeah. There are some startups in the area. There are few ones in multiple directions. So one of them that's maybe closest to what we do here is like uh, they're trying to do the CAD tools for biological for bacteria more or less, like things that we would have like when you do like an electronic circuit, you have all these CAD tools that tell you, oh, you put in your Boolean function and then it synthesizes it down to like a floor plan, and then the floor plan goes to placement and everything. They're trying to do the same thing with bacteria. Um,
present, we were discussing with this friend of ours who is a biologist in one of the best hospitals in New York. And he was telling us that, I mean, while we were speaking, to remind me the discussion that we had with him, he was telling us that for this kind of neurodegenerative diseases, they do some similar techniques, like they inject
Yeah.
some kind of DNA in some of the cells of the brain.
Right.
And then there is some reaction, and eventually this slows down the progress
Right.
of the disease, and that this is the future, for
Yeah.
this kind of diseases.
Yeah. The lots of areas that you can go with. Like I don't know exactly what you're referring to, but I think there are some actually some proteins that come from the virus that we've been using that can help. The protein 3 of the M13 virus. Yeah, yeah. and also like one other application for viruses that's really exciting is like this what I've been telling you about antimicrobial resistance, like that they tend to have these antibiotic resistances now, and because we've been using them so much, and they mutate towards the resistance, and now every bacterium is resistant. Like there's an alternative approach that's really exciting. It's like phages. So viruses attack bacteria. It's another way of killing them. And that's really active. Yeah, there's lots of interesting stuff in the area. Yes, so we did have the virus work that I quickly mentioned. So we did have like distributed circuits that communicated. It wasn't like with signal amplification. It wasn't with conjugation, but it was by sending viruses. So you compute something, you output, you send by a virus to the other, and it takes that up and does something else. We did that. 100%. Yeah, yeah. Yeah. 100%. Yeah, yeah, that's a really good question. So kind of the this field of synthetic biology where you try to reprogram cells, or mostly bacteria. It's also yeast and other cells, but the biggest part is bacteria, has been going on since like more or less 2000, year 2000, 26 years. There are lots of people doing that. Some of them are less, let's say, mathematically or physically or whatever inclined. You see that many of them do have a different background than biology, actually. So sometimes it's funny that like like most of them have more like a physics background than anything else, but um, you do see these kind of approaches, maybe not in the same form, but where people try to model something and then and go on with this. There are also like more experimentalists that just brute force their way to like getting a nice um, a nice system. But sorry, I think I lost track. What was your question again? Yeah. Yeah, of circuits. So maybe the most advanced thing that they can do is like more or less in terms of the multiple different like dimensions to look at. If you're looking at like count of gates that you can put inside of a cell, maybe around like maybe 10 inverters that you can put, um, order of magnitude. Of course, that's not like the impressive, because first you wouldn't need the inverters. Like every even multiple of inverters you don't need. Um, but I think what's interesting is that like the pure Boolean thing it's going on. It's improving. There are also things like in different dimensions where people have built like much better memory, for example. The memory that I presented to you is very kind of like a DRAM or something, where kind of you need to produce something over and over all the time, so that your signal stays up. There are also memories that are DNA based. So much of that has been enabled by a mechanism that has been found like a few years ago, that's called CRISPR, or like that's called 15 years ago. It's called CRISPR, where you can edit the gene very efficiently. So kind of you can write down something that happened to the cell by editing the genes. Like inside of your DNA, now you're noting down something. Maybe you're adding something, or you're taking something out, and that's also been really exciting. Some of the other more exciting things, like so the synchronized clocks that I've showed you at the video at the beginning with the green flashes, that has improved also over the years. Yeah, things are definitely improving in lots of directions now.
On bacteria, like relation dynamics, bacteria, what limitations are they how multiply the same kind of things? They tumor cell, how they can proliferate or die, something like that.
Yeah, yeah. So Bacteria and specifically E. coli are very easy, or the class of bacteria that E. coli belongs to are easy because it's relatively easy to introduce the DNA. Um, oh yeah, you can definitely do that. Yeah. Yes. 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 Exactly. Yeah. 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 You hit it on the nail on the head. So it's much more spatial. Whenever you're talking about tissues or something else, it's you have to be spatial. So it's a different kind of model you need there. Maybe PDE based or partial differential equations or others. Yeah, correct. Yeah. Yeah. Oh, yes. So you depends on whether you see population protocols as being part of that or that's more just clear. Some of the techniques carry over. Yeah. 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 Right. Right. So some of the things that I don't think are useful there, like I don't know, like taking some complicated algorithm and trying to implement it, like I don't know, trying to do EIG consensus or something. That's it's just too complicated. You won't be able to do it. There's some ideas that you can take out of it, and some of the ideas actually is what I've like related to something that I presented with the dual rail encoding. So if you do this dual rail encodings, you approach like something that actually the chip design community has taken, maybe not from the Botzy community, but like maybe at least adjacent, or it's like some communities in between. It's like building asynchronous circuits out of it. And then there are these synchronization or like yeah, synchronization mechanisms between like local signals that are not crazy to assume that you can implement. Like Muller C gates or something is something that you might be in terms of like, for example, like having a fixed bound t on your like a threshold on your faulty things. I don't think that's super useful because first n changes. So, um, but also like the robustness is kind of a different nature. Like the robustness is not like the Byzantine that tries to trick you up, but like it's that some of things are stochastic. Some things that you expect are happening don't happen. Some things you just don't know about. Uh, so it does have a different flavor for many things. So. Yes, you can only try to go away from you with so much. Yeah. No, but because that's serious, it's useful, like because you can implement the signal amplification. Yeah, 100%. 100%. Yeah. So there are people doing like what's called cell-free systems, taking like mechanisms out of the cell and just doing them in the tube. You can do that. Yeah. More or less like what happens there is that you would be restricted, quote unquote restricted, to the first part of the lecture, meaning that now you're just chemistry, you don't have the compartments. So compartments are sometimes a disadvantage and sometimes an advantage. Oh, yeah. Oh, yeah, 100%. So you're right. So if you're, so there are a few things. So the biggest pull that I see personally is that you can do things in vivo You can, for example, ingest bacteria and maybe they do something interesting for you. You wouldn't be able to, if you just drink the chemicals, that's not going to happen, right? And also like in terms of like bioproduction is a thing. So people do produce things with living matter. So everything is not a chemical reactor. Some things are bioreactors.